Welcome everyone, and thank you for joining today's webinar, where we'll discuss how we at InterPro are using large language models to describe protein families, and how we automate this annotation process. As Andrew mentioned, my name is Matthias Blum, and I'm the development project leader for PFAM and InterPro at EMBL-EBI. So protein annotation is a key challenge in bioinformatics. There is so much sequence data available now that traditional manual creation methods can't really keep up. So our team has been working on ways to use AI or large language models techniques to help with this workload. Unlike the two previous sessions with Andrew Green and Melanie Volmar, this webinar will be relatively non-technical. I will be talking more about how we are using existing large language models rather than explaining how to train your own. So it's more about our journey into journey of exploring and adopting LLMs. So let's get started. Sorry, let me give you a quick rundown of our agenda. First, I'll explain why classifying proteins is so important, how it helps us to understand function, structure, and drives research in medicine. Then we'll take a brief look at the history behind protein classification and why databases like PFAM were created. I'll also explain why we needed resource and we still need resources like InterPro to integrate all these databases. After that, we'll talk about how InterPro entries are normally created and the challenges we face during the creation process. This will bring us to using LLMs to automatically describe the protein families. I'll start with the two very, very naive approaches that didn't work for us. Next, I explain our initial attempt to summarize literature using LitSum. Then we'll move on to our main method, which uses Swiss-Prot data to generate protein family annotations. I will also briefly talk about our experiment with a fine-tuned model and compare its performance with commercial models. And finally, for protein families that don't have any Swiss-Prot data, I'll describe our latest approach with use gene names instead. So I know this is a lot of background, and I do apologize for taking extra time on it, but I believe it's essential to understand our challenges and why we made certain choices, or why some method works, why some method work and other don't. So with that in mind, let's jump into why classifying proteins is so important. So why do we classify proteins? Because at its core, protein classification is about understanding the building blocks of life. Proteins are made of distinct functional units called domains. Each domains bring unique properties, and when combined, they form together proteins with a wide range of functions and structures, a bit like Lego bricks that nature will put together to make different protein functions. So on the right, we have a representation of the structure of the human P53 tumor suppressor protein. This is a crucial protein that is found in many vertebrates, and it's crucial because it helps regulate the cell cycle and prevents formation of cancer. So it's composed of several domains that are represented in different colors in the 3D structures and on the 2D diagram. So the presence of a particular domain can be indicative of the function of the protein. Therefore, by identifying and classifying these domains together, we can predict the function of proteins, understand their roles in various biological processes, and gain insight into their evolutionary history. For example, if we see that a protein contains a kinase domain, we can reasonably guess that this protein might be involved in phosphorylation, a key process in cellular signaling. Beyond basic research, protein classification is also critical in more practical fields like drug discovery. Knowing which proteins belong to the same family can help design more targeted drugs and identify biomarkers for diseases. So one of the biggest challenges we face today is the volume of data. Large-scale sequencing projects are generating vast amount of protein sequences, far more that we can experimentally characterize. This is why computational methods are not just helpful, but absolutely necessary. So these tools enables us to group proteins based on sequence similarity, making it possible to manage and interpret this overwhelming amount of information. And this is not new. Swiss-Prot was created in the late 80s, and it's a curated database of protein sequences. So each sequence is manually enriched with high-quality annotations. But as sequences technology advanced, the number of protein sequences available for researchers exploded. It's exploded so much that it became impossible to annotate and incorporate all new sequences into Swiss-Prots. This is why almost 30 years ago, Trumbull was created to make these new sequences available as fast as possible, even with few or no annotations, until they were properly annotated and added to Swiss-Prot. Trumbull was supposed to be a preliminary section of Swiss-Prots, a collection of unreviewed sequences, most of which would eventually be moved to Swiss-Prots. However, after 4 years, Trumbull was more than 3 times bigger than Swiss-Prot, and it's more than 400 times bigger now. So early on, researchers recognized the need to organize data into meaningful groups. This led to the development of resources like PFAM, Prosite, and SMART. Prosite focused on identifying conserved patterns and profiles in protein sequences. PFAM uses hidden Markov models to model conserved regions of protein families, and SMART specializes in identifying signaling domains. These databases were developed because manual curation was simply not scalable. Researchers needed tools that could ultimately process large amount of data and provide reliable annotation by transferring the knowledge from the few characters of a sequence to the many uncharacterized ones. So over time, these individual resources became the building block of protein classification, each contributing a piece of the overall picture. However, this proliferation of resources also presented a few challenges. Databases like PFAM, Prosite, and SMART initially focused on describing well-known protein families and domains, which naturally resulted in some overlapping or even redundant information. But then they evolved separately with each their different methodologies and update cycles. So this fragmentation means that researchers often had to consult multiple resources to get a complete picture, which can be both time-consuming and confusing. This led to the creation of the InterPro consortium about 25 years ago. These different predictive models that we call signatures are imported from member databases and compared by curators. When 2 or more signatures are overlapping and describing the same protein domain or protein family, they are integrated together in one single InterPro entry with a unique accession number. And of course, if a signature doesn't have any counterpart in other database, it's assigned to its own InterPro entry. So here we have an example of the earliest InterPro entry for the C2 domain. This domain is described in 4 member databases: PFAM, Prosite, Prince, and SMART. And when we look at the predicted location of the domain on the sequence, they clearly overlap. So of course, this is one sequence, but the overlap in all the sequence, they match. So when this happens, InterPro curators create an entry and integrate the member database signatures in this entry. Since then, the number of member database increased to 13, and InterPro bring all their information into a single unified platform to provide a more comprehensive and robust annotation for each sequence. Another important role of InterPro is its use in UniProts. Most proteins in UniProt are unreviewed, meaning they haven't been manually created yet. InterPro plays a critical part in providing these entries with names and functional annotations, so making them more useful to researchers. So essentially, InterPro acts as a bridge between the raw sequence data and the meaningful biological insight that scientists need. And here's the page of the InterPro entry for the C2 domain that I mentioned earlier. So an InterPro entry describes one protein family or protein domain. It's made of 1 or more database signatures. There's these predictive domains that characterize the domain and family. An InterPro entry has a name, a short name, which is kind of a mnemonic identifier. It has a complete description of the function of the protein family or domain, and literature references that support this description. The name, the short name, and the description, and also the literature reference are heavily based on annotation provided by the individual member databases. So when member database provides complete pre-annotated description of families or domains, it's easy to create an InterPro entry because we can just reuse their description. But not all families and domains come with annotation, and some databases often lack description and references. This means our creators must manually create annotation by searching the literature, which is both labor intensive and time consuming. And ultimately, this slows down the integration process. So this graph shows the number of protein families and domains provided by member databases. Families and domains provided with a description are represented in green, with the one without a description are represented in red. Take Panther, for instance. Panther is one of the largest contributors of protein families to InterPro, but yet none of its families come with descriptive annotation. So in this case, InterPro creators have no choice but to write the description of the family from scratch. Again, this takes a lot of time, and because of this, creators are less likely to work on Panther families. And as a result, for a long time, Panther used to be the member database with the lowest integration rates. So naturally, the question arises, what can we do to help InterPro creators? And this was our main motivation to explore LLMs, because LLMs have been shown to be a powerful tool that can quickly and accurately summarize text. So these models can take large amount of input data and generate concise summaries that captures the essential information, so which potentially can completely transform the annotation process in InterPro. But also, LLMs come packed with knowledge, so why not try directly asking them to describe a protein family? Because after all, if I go on ChatGPT right now and ask ChatGPT what's happening at the end of the film Sixth Sense, the model is going to give me a correct and detailed answer. So maybe it works the same for protein families. And that was the first thing we did. That was the first thing we tried. We simply prompted the language model with a protein family name, and asked to generate an annotation. So for instance, we might have used a prompt like, write an InterPro style description for the leptin protein family. The interest of this method is that it's very, very simple. It requires no additional context or data and relies completely on the knowledge that the model has acquired during training. And it works. It works pretty well, actually, for protein families that have very descriptive names and that are very frequently discussed in the literature. The model generates a reasonable detailed summary. But for families that are less studied or that have very generic names, such as uncharacterized protein or express proteins, the model doesn't know. And if it doesn't know, it's not going to make it up. Well, actually it does. And this is what we call hallucinations. And even if the model output is not hallucinated, it's overly generic. And even when the summary is acceptable, the model doesn't provide any source citations. And if it does, again, there are most likely hallucinations. So that makes very, very difficult for curators to check the generated annotation, because the model output might sound plausible, but completely lack any scientific evidence. So in summary, this approach is appealing because it requires very little, but is not reliable or precise enough for all needs. To describe protein families, especially for protein families that aren't well understood yet. Okay, so we want description supported by literature, right? Well, then let's give scientific papers to the model. So we give the model a scientific paper about a protein family, and for example, if a paper describes or discusses the structure and function of a protein family, we use it as a context and ask the model to generate a summary. The results are richer and more accurate because the model has solid context to work from. The paper helps anchor the summary in real experimental and literature-based data. But there are limitations. The quality of the summary depends entirely on the paper we choose. If the paper doesn't cover all aspects of the protein family or focus too much on one variant, the annotation may end up incomplete or biased. And again, manually searching for papers and selecting the right one is time-consuming and subjective. So And moreover, if there are more than one paper, if more than one paper is needed to cover the full scope of the protein family, this process becomes even more complex and costly, both in terms of processing tokens and human efforts. So in short, this summary is not bad. This method is not bad, especially when we come across a great paper, but it's not scalable for annotating a large number of protein families. It depends too much on manual paper selection, and the potential for a bias it has make it suboptimal. But again, since LLMs are very good at extracting information from papers, in several occasions we could generate the description of a protein family from a single paper. It's just not something that can be done for thousands of protein families, because we need a more automated solution that can scan thousands and thousands of papers. Well, we're in good luck, because this is exactly what the NRS Central team at EMBL-EBI did. They faced a similar challenge, which is having vast amounts of literature, of parsing and creates vast amounts of literature about non-coding RNAs. To address these, they, and say it's Andrew, who introduced me, developed LeadSum, which is a tool that automates literature summarization using LLMs. So LeadSum first retrieves relevant paper from your PMC that mentioned a specific non-coding RNA, then extracts key sentences and condense them into a structured summary. A major strength of LeadSum, it has a lot of built-in validation mechanism. Every claim in the summary is backed by one or multiple references. So this is achieved by enforcing formatting the formatting of citations in summaries, by using self-consistent checks and iterative quality refinements. So having this multi-layered fact-checking system ensure that the content is accurate but also verifiable. So this approach has been quite successful for the RNA Central team. They had applied LeadSum to over 4,000 non-coding RNA entries, and they generate high-quality summaries that are now available on the RNA Central website. So this allows research to quickly grasp the function and significance of non-coding RNA without manually reviewing multiple papers. So at this point, LeadSum seemed exactly what we wanted, a scalable, automated method for extracting and summarizing literature to help with protein family annotation. However, when we attempted to apply this approach, the protein families in InterPro, for protein families in InterPro, we ran into a problem. Protein families accession from metadata like Panther are almost never mentioned in scientific papers, and the family names, when they do appear, they're often too generic. Again, uncharacterized protein. That's an example. This makes it very difficult to retrieve the precise information needed to automate the annotation. So although LeadSum isn't directly applicable for protein family work, I strongly recommend you to checking the LeadSum GitHub repo or their publication, because it's a great example of literature extraction that you could potentially adapt to other areas. So given the challenge we encounter with LeadSum, we turn to a different strategy using Swiss-Prot data. The basic idea here is simple. For any given protein family, we gather all the description and annotation from Swiss-Prot entries that are linked to that family, and ask the large language model to summarize the information into one unified annotation. Swiss-Prot is considered a gold standard in protein annotation because it's manually curated and provides detailed, high-quality data. So by relying on Swiss-Prot, we're not dependent on the literature where protein families, identifiers, and names are missing or too generic. So instead, we have a rich, reliable source of information that we can use to generate consistent annotations. And this approach has several advantages. So first, it ensures that the data is of high quality. So it's from a well-known resource. Second, because the data is structured and curated, the LLM can work with clear, ambiguous input, which leads to more accurate and coherent summaries. And finally, using Swiss-Prot helps overcome the problem of missing context that we face when using the LeadSum approach when applied to protein families. So here's an example of an input prompt using Swiss-Prot data. We took a set of Swiss-Prot entries associated with a protein family and extracted key pieces of information, mainly the description and function-related comments. Our prompt will start with something like, a protein family matches 3 Swiss-Prot proteins. The proteins are listed below. And then it will list each entry with its name, comments, and other relevant details, and finish the instruction for the model. Something like, with the information provided above, please describe the function or functions of the protein family in 200 words or less. One issue, and that worked pretty well, but one issue we face was that, unsurprisingly, many Swiss-Prot proteins within the same protein family often have identical or nearly identical names, description, and comments. So this redundancy means that if you simply include every entry as it is, the input prompt can become much larger than necessary, which increase the token counts without adding any insights. And the longer the prompt is, the more token we use, and the more we risk hitting the model context limit. So this problem becomes even more pronounced for protein families that have many Swiss-Prot entries. For instance, if a family has dozens or hundreds of entries, and if many of these entries share a similar or identical data, the cumulative effect is that the input prompt is just too long for the LLM to process. So one solution is to aggregate the data so that we will only pass unique names and comments to reduce redundancy. This often led to summaries not as good as the one generated with all Swiss-Prot entries, potentially because the input prompt was less structured, and that may have confused the model. Another simpler strategy that worked slightly better was subsampling. So we defined a maximum number of Swiss-Prot entries to consider, and we randomly pick Swiss-Prot entries in the family until this number is reached, and we use these ones for the prompt. That, however, was more this issue was more at the beginning of the project, and later on it got better, because with the evolution of large language models, the context windows became much bigger. Like, for instance, GPT-4 has an input context windows 8 times bigger than GPT 3.5, which GPT 3.5 was the first model we used to describe protein families. So this improvement means that we can now include a lot more data in a prompt without really worrying about exceeding the token limits. So instead of aggregating unique names and comments or subsampling, we can often pass the Swiss-Prot entries in their full UniProt flat file formats. So we have an example of that file format on the right of the slides. This format is well structured. It has been a standard for years because most large language models are already familiar with these formats. They understand very well the structure and extract the relevant information more reliably. What this means in practice is that we can simply clean up this file by removing unnecessary fields like the protein sequence, the date of creation, and then feed the remaining data structure data into the model. This allows us to pass all the valuable curated information, while at the same time ensuring that the input remains clear and coherent and easily understood by the model. And because we provide more data to the model, the summaries generated are often better. So at this point, we could generate description for protein families using large language models, but we wanted to take a step further. Generating a description is great, especially for protein families like those in Panther that completely lack curated annotations. However, to automatically create a full InterPro entry, we need more than just that description. As I mentioned earlier, an InterPro entry isn't complete without several key elements. Specifically, we need a signature to integrate. That might come from PFAM, Panther, or other member databases. And in addition, each InterPro entry requires a unique name and a unique short name. So for instance, you might have an entry that has the unique name bacteriophage transmembrane helix and a short name TM for transmembrane _ bacteriophage. Finally, of course, we need a detailed description, which is essential because it provides context and describes the function and characteristics of the protein family. So to automate the creation of these complete entries with name and short name, we extended our LLM approach. Instead of just generating a summary description, we instructed the LLM to produce a JSON object that contains all the required elements, the unique name, the short name, and the description. Having a JSON output also has a big advantage because it simplifies downstream processing and integration. With a valid JSON object, our pipeline could easily parse and extract the necessary fields to ultimately create an InterPro entry from scratch. This means that in theory, we will have an end-to-end pipeline that will take an annotated family and transform it into a fully described InterPro entry without requiring any human intervention in middle. However, this introduced another problem because yes, generating struct JSON ensures that we have all the necessary components, but InterPro has curation guidelines, and getting the model to consistently follow those constraints was more difficult than expected. Yes, InterPro has strict curation rules that must be followed for every entry, no exception. So for instance, for each entry, each entry needs a unique name that is 100 characters long or less, and a short name that is 30 characters long or less. And the short name must only include alphanumeric characters and a limited set of special symbols like a hyphen or underscore. And there are more curation rules. So to help the model with these rules, we include them in the prompt and use few short examples to show what we were expecting. So this way, the model had a clearer idea of how to generate names and short names that met our criteria. And in practice, most of the time, the model did a fairly good job following the instruction. However, there were still occasions when it produced names that were either too long or had invalid characters. And sometimes the generated names ended up being duplicated of existing entries, which again is not allowed. And that occurred especially for protein families that hit a wide variety of proteins, which led to more generic names. So in this case, we could still display the generated description on the protein family page on the InterPro website, but we cannot create a new InterPro entry for this family. We also thought about using a retrieval augmented generation approach, or RAG. With RAG, the model will pull in information from a database of existing InterPro entries to ensure names are unique. While this sounds good, we haven't used it yet. And one of the reasons is that there is a risk with forcing uniqueness. If we do this, the model might prioritize generating novel and unique names, but less semantically correct. So overall, getting the model to stick perfectly to all rules takes a lot of trial and effort, and even repeating the instructions several times in the prompt doesn't always guarantee perfect result every time. So it's kind of like an art or witchcraft. It's difficult to really have the model to follow your instruction 100% of the time. Still, we could generate annotation for over 5,000 protein families and use nearly 4,000 of these to create new InterPro entries and make them available on the InterPro website, in the REST API, and in the files we distribute on the EMBL-EBI FTP sites. Of course, we always clearly indicate when annotations have been generated using AI or large language models, and user can provide feedback. And when we receive feedback, usually it's pretty good. It's pretty positive. So this chart shows how the number of InterPro families has grown over time. It really highlights how AI or LLMs helped fill annotation gaps in the past 2 years. Currently, AI-generated entries account for about 15% of all InterPro families available on the website. And at the current state of manual curation, it would have taken more than 5 years to annotate the same number of families that were created using large language models. One important thing to understand about our AI-generated entries is that they're not created and left to gather dust. We updated our internal curation application so that curators can review and approve these annotations or even modify them if needed. Sometimes the set of sequence that define a family might change over time, making the original description less relevant or accurate. That can happen for both curated and AI entries. The difference is that with AI, now we have an option to move an outdated entry into a special queue instead of deleting it. And this way we can regenerate and updated annotations instead of just losing this family. On these slides, you can see 2 examples of AI-generated descriptions. On the left, there is a description that is exactly as it was, as the large language model produced it, while on the right, the description has been updated by a curator. The curator rewarded a few sentences to make it match the InterPro style and added reference to specific publications. So what's important is that we don't expect the AI output, the large language model output to be perfect from the start or to be perfect all the time. We think of it more like a boilerplate, similar to how software frameworks like Spring, Django, React, and so on can generate starter code for a new project, which the developer then uses. Now curators have similar tools for creating basic description of a new protein family, and then they can improve this description and enrich it. So this saves a lot of time compared to writing from scratch and makes the curation process more efficient overall. One of our concerns early on was how to manage the cost of generating description with commercial models like GPT-4 or other commercial models. It's feasible to do this for a few hundreds or even a few thousand of families, but we wondered what would happen if we needed to re-annotate 20,000 families in InterPro, especially if we wanted to do it frequently, say every time a family gains or lose a sequence. That's a lot of API calls, and the cost could become prohibitive. So to explore an alternative, we looked into fine-tuning a local model. So we could generate this description in-house on the EMBL-EBI cluster rather than relying on the OpenAI API. We used a model called Sci-Fi Mistral, which is already fine-tuned from a 7 billion parameter called Mistral 7B, and it's been fine-tuned on scientific text. So the fact that it's oriented towards scientific data or scientific publication makes it a good candidate for describing protein families. Our fine-tuning strategy was pretty straightforward. We took description previously generated by GPT-4, and we used them as training data for the Sci-Fi Mistral model. So the idea was we were trying to hope that the local model, where we were hoping that the local model would learn to produce summaries that were nearly as good as GPT-4, but for a fraction of the cost, of course. And after fine tuning, we ran an A/B test to see how those local models compare to GPT-4. We generated description for about 60 protein families using GPT-4, the base Sci-Fi model, and our fine-tuned version. We then show pairs of description to curators without telling them which model produced which description, and ask curators to decide which one they preferred. If you're using tools like ChatGPT, you might have seen something similar. Sometimes you get two separate responses side by side, and you have to pick which one you prefer or which one you find the most accurate and readable. And the results are shown in these pie charts. GPT-4 was still preferred overall, but the fine-tuned Sci-Fi model was not far behind. This means that we can use local models to generate high-quality protein family description, especially if they're fine-tuned on data from strong model like or from curated data. At the moment, we still mostly use OpenAI models because they strike as a good balance between cost and quality for our current workflow. But if we were to systematically regenerate description for all InterPro families on a regular basis, we'll most likely turn to a local fine-tune model. So far we're using Swiss-Prot data to annotate protein families. Other using the standard the OpenAI models or a fine-tuned local models, and it works pretty well. What happens if we don't have any Swiss-Prot data? There are many protein families that don't match any Swiss-Prot entries at all. And this is because Swiss-Prot is a very small subset. Summaries is much bigger than Swiss-Prot. So yeah, we do have about 5,000 families that don't match any Swiss-Prot. We have and that's a question: how can we address this? So we developed a method that uses gene names instead of Swiss-Prot entries. The idea is that even if we don't have Swiss-Prot data or a very small amount of Swiss-Prot data, we can retrieve description on genes function from the literature, and that should provide enough context to create a good description. But again, the issue is that protein families can match a very high number of uniprots because Trumbull is so big. So we have to apply a filtering step to only retain the genes that are the most specific to the family. That's a good slide. So once we have that list of gene names, we need to find which ones are truly specific to the protein family of interest. And to do that, we retrieve all proteins from UniProt for each gene, and for each of these protein, we look at the protein families matching their sequence to know if a specific sequence belongs to the family, a protein family of interest, the one we're trying to annotate, or to another family. So we then calculate the proportion of sequences that belong to the family of interest versus the proportion of the number of sequences that belong to different families. And if a gene mostly encodes protein associated with other families, we consider it to generic and we discard it. This filter step really reduces the number of genes and therefore the number of families that we can annotate this way, but it really improves the reliability of our annotations, and that really ensures or guarantees that the gene names are truly relevant to the family of interest. After filtering out non-specific gene, we get to the snippets retrieval stage. So again, very similar to LeadSum, we search open access articles in Europe PMC to find any paper that mention that genes. And we're gathering the sequence that talks about the gene function, expression, and other details. And the idea is that these extracted sentences or snippets can provide context that we need to build the annotation. Once we have collected the snippets for each gene, we summarize them again using large language models. And the goal is to get for each gene a concise, coherent description of the function of the gene based on the literature. Really going quickly on the polysemy thing. So the idea is that some gene names have, share are the same as common words like arm or Inca, and that can bring false positive from the literature. So we have a few system or mechanism to try to filter out or ensuring that snippet is really about a gene function and not about something completely irrelevant. But there are some cases where the ambiguity is the risk of ambiguity is too high, so we just discard the gene. And when we have one summary for each gene, we combine them together to form the final family description. So we prompt the large language model again, but this time use all the individual gene summary, and we instruct the large language model to provide a description for the entire family. And yeah, we get a complete description. And this time, because we based the data on the literature, each statement, each sentence in the final description is supported by literature, and we have the citation or the reference to the literature in the description. So I think I stopped just there before going back. So like for the fine-tune model, we wanted to do an evaluation to compare the description generated using gene names with the description generated by Swiss-Prot entries. But this time we also wanted to compare the description generated by human curators. So instead of generating description for families without any annotation, we picked families that already had annotation written by curators in the past. And again, we did the same thing as we did in the other evaluation. So we show pairs of description to curators and ask them to choose which one they preferred without telling them the method that was used to create the description they were comparing. Since description generated by human curators or expert curators or using the gene approach can include literature references, citation to the literature, we remove them for this evaluation, so curators will not know which method was used. And the pie charts on this slide show the results. So when comparing a description generated automatically using Swiss-Prot data with a description from the same family written by a curator, in 75% of the time, curators prefer the one generated by LLM. When comparing a description generated automatically using LLMs, but using the gene using gene names with a description generated by curator, curators prefer the one generated using gene names, so the automatic one, in 56% of the time. And when comparing two AI-generated description, one using Swiss-Prot data and one using gene names, curators prefer the first one, the Swiss-Prot one, in 62% of the time. So the final ranking is Swiss-Prot data on top, gene name second, and human last. So this shows that LLM-based annotations don't just rival with human written description. In terms of clarity or completeness, they are often better. The Swiss-Prot based method is typically the most accurate and up-to-date for well-characterized families, and the gene-based method serves as a nice or decent backup when Swiss-Prot data is not available. Before we wrap up this presentation, it's important to highlight one a limitation in our methods. In the introduction, I talked about protein domains and protein families, but then I stopped mentioning domains. It's because Swiss-Prot description and gene-based snippets taken from the literature typically describes the protein as a whole, so the full sequence of a protein. However, protein domains, repeat motifs, and other subregions can have distinct functions and characteristics that are not necessarily captured by the full-length description. So as a result, our methods work pretty well to annotate protein families, but they are less suitable for generating domain-specific annotations. So this is something we want to explore in the future, because there are protein domains that lack like protein families descriptions. So here are some take-home messages and next step for our LLM-based annotation work. LLM have proven to be a powerful tool that complements manual curation. When sufficient data is available, the Swiss-Prot approach yields the most accurate summaries, and often better than what expert curators wrote. And the gene-based approach is good for families lacking Swiss-Prot entries. Looking forward, we need to keep improving our prompt templates to generate annotations that follow InterPro style and rules better. We also want to keep exploring local models, as very good models are released almost weekly, and benchmarks show that they are getting closer and closer to commercial models. Haven't talked much about hallucinations. So we saw a few when using Swiss-Prot entries and a bit more when using gene names. So we need to develop an automated fact-checking system to reduce or even remove, if possible, these hallucinations. And finally, we started to look at combining methods, like feeding Swiss-Prot entries and gene names to the LLMs. So this is promising, because so far we saw that when combining different sources of data, Swiss-Prot entries, gene names, or gene literature snippets, the generated annotations are even better. So this is something we want to explore. And with that, yeah, thank you for your attention. I'm sorry for the slide issue, and I'm happy to answer any question.
Thank you very much, Matthias. So there's a few questions in the Q&A. So the top one at the moment is about a limitation with GPT-4 and similar models, that they don't tokenize gene symbols properly. So they will split a gene symbol into multiple tokens. So the person is wondering if we've considered using a gene or protein specific tokenizer or any kind of approach like that.
We haven't, but it's a great idea. There are a few papers that mention this. So far, so this gene-based approach was our rescue system that was implemented not in a rush, but to save those families that couldn't be annotated using Swiss-Prot. So the vast majority of annotation are being generated using Swiss-Prot data. We don't have so many entries that have been created using the gene-based approach. So that wasn't our main focus, but yes, that's something we pretty need to do.
Thanks. So the next one is asking, could you give an estimate of what this cost? So what did we spend doing all this?
Okay, so if we remove the trial and error, because sometimes you keep regenerating the same annotation over and over, expecting to have something stable, playing with the temperature. But if we had the pipeline we have right now in production and run it from start, I would say between 1,000 and 2,000 pounds. It's a wide range. It's a pretty wide range, but it's less than 2,000 pounds.
Okay, thanks. Refresh this. So have we thought about putting any of the curator feedback back into the model as a kind of reinforcement learning sort of loop?
Let's say that would be nice. That would be nice, but that means we need to not force, but encourage more creators to look at the AI generated entries. And with few curators available, or not a lot of curation available, time available, usually they are more focused on creating new entries or domains. But that's a good idea. The same idea to a similar idea would be to use the existing description written by curators and that are considered good to reinforce the learning of the model.
Okay, and then there's a question about contradictory annotations. So what do you do anything to work out what to do with those, or what how can you tackle that?
No, at this the moment we when we do see that, we just flag them as to be removed, so they won't appear on the website anymore. But they go on a queue of to review. So yeah, we don't give bad information to users, but yeah, so far we haven't done anything with those.
Okay. You mentioned you want to try and detect hallucinations. Have you got any ideas how you would plan to do that?
Well, a similar approach to so if when using literature, like for the gene base, we can apply a similar approach to what you did for lead sum, which means that for every single sentence says, look back at what I gave you. Is anything in this input supporting what you generated? That's one of the possibility. But for Swiss-Prot data, it's more difficult. You will need to have to feedback again the entire Swiss-Prot file and say, look again. So the model can always say, yeah, sure, of course, everything is supported. So eventually, without spending a big amount of time with curators, it's difficult.
So we've got a couple more minutes, so we'll do a few more. Have we tried using the newer generation of reasoning models on any of this sort of task?
I believe that some of the latest annotation generated using the gene approach have been used using the reasoning models, and they are good, but they're not significantly better than the other one used with the generative use with non-reasoning models. But that I wouldn't say it's because the reasoning models are not suitable. It might just be because the papers introduce a bias. Maybe those papers are less descriptive, or the protein family is more generic. It's something we should try with Swiss-Prot ones and the description generated with Swiss-Prot, and then compare those on a small subset of 100 and asking again first to evaluate those.
Okay. And then I think one last one, because I think we're running short on time. Have you considered using LLMs to review the existing summaries and scan for outdated or mistaken entries to then update them?
Yes, that is pretty one of our priority for 2025. But we have 20,000 protein families in InterPro, and the oldest one are the one hitting the highest number of Swiss-Prot. So again, either we'll go down the road of subsampling it, or we'll have to find a model that has a huge context window. But if we were to go through these 20,000 and use a commercial model like OpenAPI, that cost would be much higher. So yeah, that will take some time, but we'll end up using a local model.